Hemostasis is a process that can not only stop bleeding, but halt the loss of blood through the walls of damaged vessels. It can also set the foundation for a framework for tissue repairs. In this video, we will be discussing the 3 stages of hemostasis, which are the vascular stage, the platelet phase, and the coagulation phase. Additionally, we will be touching on clot retraction and fibroanalysis. First, the vascular phase of hemostasis can be triggered when the wall of a blood vessel is cut, thus triggering a contraction in the smooth muscle fibers of the vessel wall. This contraction of the vessel is called a vascular spasm, which decreases the diameter of the vessel at the site of the injury. During this phase, the constriction can slow or stop the blood loss through the wall of a small vessel. This vascular spasm lasts about 30 minutes and is the vascular phase. During this phase, endothelial cells contract and expose the underlying basement membrane to the bloodstream. The endothelial cells can also release chemical factors, local hormones, and peptide hormones called endothelins. The endothelins stimulate smooth muscle contraction and promote vascular spasms, and stimulate the division of endothelial cells, smooth muscle cells, and fibroblasts to accelerate the repair process. Lastly, in the vascular stage, the endothelial plasma membranes become sticky in order for small arteries or veins to be sealed and to aid platelets in attachment, which brings us to the next phase, the platelet phase.
The beginning of the platelet phase starts when the platelets attach to sticky endothelial surfaces, the basement membrane, and to the exposed collagen fibers. This is also known as platelet adhesion. After that, the platelets release the compounds adenosine diphosphate, or ADP, thromboxane, clotting factors, platelet-derived growth factor, calcium ions, and all this starts the process of platelet aggregation. This is when more platelets arrive and stick to each other. ADP, thromboxane, and calcium ions from each platelet stimulate further aggregation, creating a positive feedback loop, which creates the platelet plug that is further backed up as clotting occurs.
When the epithelium tissue is broken, the first thing that happens is platelets come and form a plug into the broken endothelium tissue. Okay, stop. So now this platelet plug is not strong enough to hold it on its own, so it needs these fibrin strands to come in and help keep it together and form a mesh over this platelet process. And so now fibrin, these strands of fibrin, these come from these fibrin molecules that piece together. And since these fibrin molecules are attracted to each other, it forms these strong fibrin strands. So now the problem is, since these fibrin molecules are all attached to each other, they're really sticky. And so when these guys are flowing through the bloodstream, we can't just have them stick together. So before they even turn into fibrin, they have a piece that covers their active site, just like that. And this is called fibrinogen. And then later on, this fibrinogen turns into fibrin, which then turns into fibrin strands. Well, the big question is, how does fibrinogen know to turn into fibrin, stick together, turn into fibrin strands, and cover these platelets? Well, the big key component is that when this endothelium tissue breaks, all these little proteins are released. But all these little proteins, they don't just work together and send signals. What actually happens is this big key factor called thrombin. It comes in, and that is the key player that tells fibrinogen to turn into fibrin strands. Okay. So now the different factors to get to thrombin, to get to fibrin, is it's going to start with 12. And then from 12, it's going to go to 11. And then from 11, it's going to go to 9. Don't know why it goes in this order, but it does. And then from 9, I'm going to go to 10, to thrombin, which is also called 2. From thrombin to fibrin, which is also called 1. But there's obviously numbers missing, and so instead of putting them in order like normal people do, we're going to put them in little branches off to the side, because I guess these little proteins work better together. So 5 is going to go with 10, 8 is going to go with 9, and this entire process right here, this is called the intrinsic pathway. This is the intrinsic pathway, and it's not that factor 12 is turning into factor 11, which is turning into factor 9. It's more that factor 12 is acting as a catalyst to activate the inactive form of factor 11, which is then activating the inactive form of factor 9, and so on and so forth until we get to thrombin, and then we get to fibrin. Okay, so this extrinsic pathway, it starts with a spark, and this spark, this factor 3 right here, this like initial spark, that comes from these protein guys from the very beginning, from the endothelium tissue damage. So that sparks this extrinsic pathway. So it starts factor 3, which activates factor 7, which then activates factor 10, which then activates thrombin. And then thrombin, once that gets initially activated, it's going to then activate 5, activate 7, activate 8, activate 11, and then activate factor 13. Okay, and then the last factor that it activates is factor 13. What is factor 13, you may ask? Well, eventually we want this fibrin to turn into fibrin strands, but we also want them to stay together and have this mesh lining to keep these fibrin strands together. And this process right here, this is this factor 13. So thrombin is activating factor 13, which is these mesh strands forming together.
Clot retraction is a process that reduces residual bleeding and stabilizes the injury site by pulling the torn edges of the vessels closer together. This in turn reduces the size of the damaged area, making it easier for fibroblasts and smooth muscle cells to complete repairs. This then leads us to our last topic, fibrinolysis.
While the vessel continues its repair, the clot starts to dissolve in a process called fibrinolysis. This starts when the two enzymes, thrombin and tissue plasminogen activator, are released by the damaged tissue at the injury site. These enzymes activate proenzyme plasminogen, which makes the enzyme plasmin. Plasmin digests the fibrin strands and erodes the clot.